about Rarity, Carnivory, and Promiscuity.
Oh.
All right.
It's
There
an
we
honor to be here with all my Miami friends and Miami mentors. And I have to tell Jim, the favorite class I took with him was plant taxonomy. And we drew all of our families, and I saved all my drawings. And now when I teach plant taxonomy, I make my students do all the drawings. And I show them my pictures. So thank you, Jim. One other thing, he would always have extra credit. I would get 102 and 106 and 109. I get 99 on my paper. I was like, Jim, why? He said, well, my grade book only goes to 2 digits. Not fair, Jim. I survived. All right, so I'm excited to talk to you about some of the work I've been doing on these rare insectivorous plants in the fall line sandhills in west central Georgia. So fall line, this is where Piedmont meets coastal plain. And you have this nice series of gentle waterfalls. So that's what's a fall line. This is also the ancient coast in the southeast. So this is where we have massive sand dunes, some of them 400 feet deep. And as the ocean was waxing and waning over those times, you get layers of sand and layers of mud and layers of sand. And so you have these interesting hillside seeps, and all these interesting plants end up in this environment. As the ocean retreated, all these things were kind of left there, kind of stuck there. So we have very unique combinations of different plant species in these fall line sandhills. So our goal was to characterize these wetlands and determine if we had any new plant communities. And so we were comparing to what's on NatureServe and in the Natural Heritage Program for Georgia. We are documenting these species using traditional taxonomy. We also did some DNA barcoding, trying to build a barcode library of the fall line sandhills area, both the uplands, the dry areas, and the wetlands. We were also looking at the hydrology of these sites and the soil types. Another goal was to develop some management recommendations. Historically, fire was very common on the landscape in the southeast, and that's been removed. So you have a lot of woody vegetation moving in. We've been working with Fort Benning, which is just south of Columbus where I'm at, the Department of Defense, private landowners, Georgia Power, because as Georgia Power puts in power lines, they open these areas up, and then you get these incredible plant communities. The downside is the way they keep those areas open. They don't use fire, they use four-wheelers. They go in and tear up these sites. We've been working with Georgia Power and the Department of Defense to come up with some strategies for maintaining these sites and protecting these sites. So, okay, there's Georgia, west central Georgia right here. This is the fall line sandhill that runs through the state, goes on up into the Carolinas and then back through Alabama and Mississippi. This is Fort Benning. This is where we started our project. And in the past, probably 10 years, Fort Benning has been purchasing thousands of acres. So this is 180 some thousand acres. They've been purchasing thousands of acres to the east in the sandhills. This is called the Army Compatible Use Buffer Zone. And so now we have access to all these new sites that were privately owned. And those landowners that were not interested in selling, Nature Conservancy has worked with them to put conservation easements on those sites to protect these plant communities. And the way we were selecting our sites, we were using aerial photography and looking for bog areas. We would go in and ground truth and then determine if we wanted to set up a site there. We had 11 sites. We followed the Carolina Vegetation Survey method. Nomenclature follows weekly. 2015, we did collect voucher specimens and we collected samples for DNA isolation and amplification. We collected soil samples looking at percent organic matter, pH, hydrology, the water content. We also estimated the landform index. So it's very different if you're standing on a hillside seep versus a beaver bog where you have a pool that's holding a lot of water. And then we were always estimating percent cover of water. This is what one of our typical plots would look like, a 20 by 50. And within that plot, you have these 10 modules. In modules 2, 3, 8, and 9, we had intensive plots within, and then there was nested subplots. So within those plots, every single stem was identified and then given a cover class. And then once we finished with those intensive plots, then we would walk the entire site and identify any other species we hadn't already recorded and give them cover classes. So we were looking at herbs, shrubs, trees, and then percent cover for each of those species. We analyzed our data looking using cluster analysis. We used PC-ORD. Then we also use canonical correspondence analysis to determine those environmental influences on those species composition. I mentioned DNA. We have hybridization of 2 pitcher plants. One pitcher plant is common in our area. The other one was not previously recorded, and we found this at several of our sites, this species at several of our sites. And then we also found hybrids. And then these are both graduate student projects. We had another graduate student building a barcode library of the fall line sandhill plants. The goal is to use this information. One of the reasons, I'm sure I shouldn't record this, but one of the reasons Fort Benning has been purchasing land is gopher tortoises are about an eyelash away from being listed. If they become listed, then Fort Benning it will be unusable for training. They're going to have to move those gopher tortoises. And so understanding the preference, the diet preference of those gopher tortoises, if we have a library of the plants we find in the sandhill, we can look at gopher tortoise species and determine what it is they're eating and what they prefer, and then enhance those environments. So that's part of our goal. Right, so this is what one of our sites look like. I'm okay with water. I'm very comfortable in the water. What I'm not okay with is mud and water. One of my fears is getting stuck in this mud and drowning. So this really pushed my comfort zone. In addition, of course, we had cottonmouths at our site, and we had wasps. Wasps are actually worse than the cottonmouths. So when we looked at cluster analysis, our sites, this is based just on species composition, our sites clustered looking at wetland seeps, and then these deep mucky mud sites, and then open water sites. When we looked at those environmental variables and applied that, it does look like we may have a new plant community, and it's most similar to what's called G1. This is your global ranking, so it's critically imperiled. This is the East. It's similar to the East Gulf Coastal Plain Muck Bog Association. That's not easy to say. And it does look like the percent water cover, the landform index, soil pH, and then organic matter. Those are the things that are really influencing those species compositions. So the new bog, this new habitat, is characterized by having high water cover. We do have some herbaceous indicator species, things like Sarracenia, Drosera, Xyris. If you know anyone who is an expert in Xyris, I hate it. It's a pain in the butt. And then there are these little woody vegetation, little islands of woody vegetation where we had Morella, Nyssa, and Lyonia. 230 different species, 110 genera, 69 families. 15 of these were rare to the state of Georgia, and 9 of these plants were carnivorous. And I'm going to talk about some of these different discoveries, but I want to point out the Sarracenia psittacina. This is the parrot pitcher plant. It's known in Georgia, but about 100 miles south of our sites. So we extended the range significantly northward. And this is a pitcher plant that was hybridizing with our more common pitcher plant. We also found Ericolum texensis, which is one of the bog buttons, and it was formerly restricted to the outer coastal plain. So these are some of the major finds. And I'll talk a little bit more about those. All right, so the red milkweed, again, G is your global ranking, so these are not impaired globally. SH is state historical, so this was thought extirpated from the state. We found this milkweed at 3 different sites. Again, these are big plants. They're over 5 feet tall, huge pink blooms. But people in the right minds aren't going out to these sites, so that's why they haven't been recorded. I'm convinced of this. Northern white fringed orchid. We had 2 plants at one site. This is one of those this is one of the hillside seeps, and Georgia Power contractors drove their ATVs through our site, destroyed one of the 2 plants. They got stuck. You could see they got stuck and they tore it up. So this is one special concern, and it's really sad to see our one plant messed up. So this Carex exilus, coastal sedge, this was a new species for Georgia, never been recorded in the state. I wanted to mention the Atlantic white cedar, and you notice this is the fall line Sandhills right here. This is our area, Fort Benning, and this is probably Fort Gordon up here. And those military installations inadvertently preserve extensive species, different habitats, animals, and plants. So it's really interesting to live that close to a military installation. Anyway, these Atlantic white cedars, most of these populations were owned privately. Working with Nature Conservancy, many of these are now under conservation easement, so they are protected. I wanted to mention the Pinguicula. This is, again, we're looking at these area photos. We can see a nice site. We know it's there, but we can't get to it. We walked along the stream. We found a little tiny opening in the woody vegetation. We pushed our gear ahead of us and we crawled to this little opening, and it opened up into this shallow, sandy, braided stream just covered with Pinguicula. This is probably the best population in the southeast. Some the other insectivorous plants, a couple species of sundew, and several species of bladderworts at our sites. I mentioned the molecular work. These are both graduate students. We did confirm that we do have hybrids between the Psittacina and the rubra, the 2 pitcher plants. And then again, the idea of building this library, this barcode library of the fall line sandhill flora. Back to the pitcher plants. Using RBCL and microsatellites, Christina Caldwell was able to determine that both haplotypes in the hybrids were present, but she was able to also see that the hybrids were preferentially back crossing with rubra. Rubra is the pitcher plant that's more common in our area. Again, Psittacina is the one that we extended the range northward by about 100 miles. And again, this is what Psittacina looks like. Typically in open water, it lays down in the water. Rubra is close to the water, not usually in it, stands up. And then this is what the hybrid looks like. This is one of the morphotypes of those hybrids. And you saw every range that you can imagine in morphology. She had over 300 specimens of the parent plants and the hybrids. Scott Silvas was working on the DNA barcode library. Using RBCL, he only got 62% species resolution. This is probably because 44% of our genera are polytypic. So the goal is to add other markers in there and further resolve those species. So again, we want to do more genetic work. We are continuing the taxonomic work. Like I said, I've got everything but Xyrus. That's one I can't get. Michele and I would like to add 2 or 3 additional sites similar to the East Gulf Coastal Plain muck bog, but that's easier said than done. It's not even easy to say. We again want to develop reference conditions for this new combination of plants. We would like to have a restoration site on one of the areas that is now owned by Department of Defense, put some raised walkways in so individuals can go in and look at the orchids and all the insectivorous plants. And then again, it's really important that we develop those management recommendations working with Department of Defense, Georgia Power, and private landowners. So Midwest Vaco, Fort Benning, and Columbus State University provided funding for these projects. These folks were at Fort Benning. Kevin Burgess is one of my colleagues at Columbus State. Christina and Scott were graduate students. And Brad Wilson is actually a veterinarian from Atlanta who just likes going out into bogs. Any questions?